Our units on biochemistry and cell structures will really come together in our next unit on cell communication. And this is video 7.1, which will introduce you to the basics of cell communication and look at one family of cell communication pathways, G-protein linked signaling pathways. We'll cover these first few objectives, so pause if you'd like a closer look. Cell communication, or cell signaling, is just what it sounds like. One cell, a signaling cell, like this cell to the left, sends a chemical message to a target cell, like this cell on the right, to elicit a response or change in behavior. The chemical message used for communication is called a signaling molecule, or ligand. The ligand binds to our receptor once the ligand reaches the target cell. One thing to remember is that not all cells will have the receptor for every ligand. Only the cells that need to respond to the signaling molecule will have the appropriate receptor. And you can see in this example diagram, only this cell in the middle can respond to the signal from the cell on the left. The cell on the right can't respond to the ligand because the cell on the right doesn't have any receptors for the ligand. Now, local signaling happens when cells are very close to one another, and you can see some examples here. In this panel on the left, the cell in the middle is the signaling cell. The signaling cell is releasing the red ligand by exocytosis, and the red ligand binds 3 nearby target cells. Since the target cells are so close by, and this is called paracrine signaling. The middle panel shows local signaling between 2 neurons. The neuron on top is releasing red neurotransmitter ligands by exocytosis of these vesicles. Then the neurotransmitters diffuse the short distance across the synaptic cleft and bind to receptors on the target cell. And finally, the panel on the right shows us that cells can signal each other by touching too. In this case, the cell on the left has membrane-bound ligands that are green that touch the purple membrane-bound receptors on the target cell. Cell communication can also take place over long distances. Signaling over long distances is called endocrine signaling and involves the signaling cell releasing hormone ligands into the bloodstream, and then the hormones travel all over the body but only bind the cells that have the right receptor on the target cell. Regardless of if the signal is local or long distance, every cell signaling pathway has 3 big steps to keep in mind: reception, transduction, and response. These 3 steps describe what's happening in the target cell. So first is reception, and reception is simply when the ligand signaling molecule binds its receptor on the target cell, and usually ligand binding activates the receptor protein. Next is transduction, when the activated receptor transfers the signal from outside of the cell to inside of the cell. Now, typically, transduction involves helper molecules called relay molecules. Finally is the response. Why the signaling happened in the first place. As a result of receiving the signal, the target cell responds by changing some activity. A response can be something like a change in the chemical reaction of a target cell or turning a gene on or off in a target cell. It's easier to imagine these steps when you have a diagram, so let's check out this picture right here. This diagram shows a target cell receiving a signal from another cell, and the first step involving this target cell is reception taking place on the left. So you can see the red ligand signaling molecule fitting into the receptor. Now, interestingly, this interaction between the ligand and the receptor is specific, meaning that each ligand has its own specific matching receptor. And not only do the ligand and receptor shapes fit together in a complementary fashion, but the biochemistry between the two has to be complementary as well. Anyway, ligand binding to the receptor causes a shape change in the receptor protein that triggers the next step in the process. Even though receptors are always proteins, you'll find that receptors can be found on the membrane, like we saw in that last picture, but sometimes in the cytoplasm too. Now, for ligands to reach cytoplasmic receptors inside of the cell, the ligands have to be steroid hormones, like testosterone in this example. And since steroids biochemically are hydrophobic and they're derivatives of cholesterol, those steroids can diffuse right through the membrane to get to the cytoplasmic receptor protein and bind and activate that protein, and then you can see this protein causing a change in DNA transcription. Okay, so let's go back to our original diagram. So the ligand binding and shape change of the receptor protein triggers the next step of transduction, where the signaling process is now brought into the cell. So you can see that transduction involves these blue relay molecules, and often there are several relay molecules arranged in a chain, one right after another. After the signal is passed down the chain of relay molecules in transduction, the target cell will change in some way, and this change is called the response, and can range from a change in gene expression to a change in a chemical reaction, and even other things. Now, besides the 3 steps we just saw, signaling pathways have to have a way to shut down, and that's called termination. And most pathways have steps for amplification. Amplification can take this one receptor ligand binding event and turn that into many downstream events that get amplified along the way. So this is why there's so many relay molecules usually, where the signal gets more and more intense, more and more amplified with each step of the pathway. Now that you have a general sense of how signal transduction pathways work, I want to get more focused by looking at a signaling pathway family. 3 pathways that we'll look at are G-protein linked receptor pathways, receptor tyrosine kinase pathways, and ligand-gated ion channel signaling. Now, don't freak out. All of these pathways still use those same basic steps, reception, transduction, and response, as well as the same basic parts, a signaling cell, a ligand, a target cell, a receptor on the target cell, and relay molecules. So for the rest of this video, we're just going to zoom in and take a look at G-protein linked receptor pathways. G-protein linked receptor pathways get their name because one of the relay molecules is a G-protein. Check out the G-protein in this example that's in the middle of all of the action. G-proteins are proteins that are active when bound to GTP and inactive when bound to GDP. That GDP-GTP molecular switch is how G-proteins got their name. See how the G-protein in these diagrams is inactive and bound to GDP at the start? Then that G-protein gets activated when GTP binds in the 2 middle panels, but then turns off again when GDP is bound. G-proteins use GDP and GTP as a molecular on-off switch. In addition to the G-protein pathways still have our old friends, a ligand right there in red, and a receptor shown in purple. And another characteristic molecule that G-protein pathways have is an enzyme, shown here in blue. In G-protein pathways, the enzyme and receptor are often integral membrane proteins, while the G-protein is a peripheral membrane protein. So now that you've met these main players, these structures, let's dig into how the molecules work together to create a signal. Well, the first step of every signaling pathway is reception, and we could see that in panel 2 right here. So just like we saw before, the red ligand binds to the purple receptor up here, and ligand binding causes a shape change of the receptor from its inactive to its active shape, so now the G-protein can attach to the activated receptor. Transduction can be considered to start the moment the G-protein binds the activated receptor. And you can see here when the G-protein binds to the receptor, a change in shape of the G-protein is triggered. This shape change pushes off GDP and creates a spot for GTP. And then once GTP binds to it, the G-protein is considered activated. Now, what does this activated G-protein do? Well, you can see in panel 3, the activated G-protein will leave the receptor and bind to the enzyme. And G-protein enzyme binding causes a shape change in the enzyme that you can see from the inactive enzyme shape to the active enzyme shape. So now the enzyme is activated. Once the enzyme is activated, enzyme's going to do what enzymes do. The job of enzymes is to increase the rate of chemical reactions. So now that that enzyme is activated, a chemical reaction can speed up, and this could be considered the response phase of the signal in this diagram. Well, what about termination? How does this pathway turn off when the signal is received? Well, check out what happens to this G-protein in panel 4. The GTP is hydrolyzed to GDP. There's that third phosphate getting kicked off right here. And this causes the G-protein to change shape back into its original inactive shape. As a result of that G-protein shape change, the G-protein doesn't bind the enzyme anymore, and the enzyme changes shape back to its inactive conformation, its inactive shape. In addition to the shape change of the G protein and enzyme, the receptor gets inactivated as well, right here in panel 3. This actually happened a little while ago. So the ligand leaves the receptor. Unbinding from the ligand causes the receptor to change shape back to its inactive conformation. So there's been inactivation of the G protein, the enzyme, and the receptor as parts of termination in the signaling pathway. Wow, that was no joke. Welcome to signaling, right? Don't worry, we've got lots of opportunities for practice, discussion, and mastery of this material in class. But let's recap the big points of this video. Cells communicate for a lot of different reasons, and they can communicate locally over short distances or over long distances using endocrine signaling. Regardless of the distance, all cell signaling pathways share some common parts: a signaling cell, a signaling molecule, a target cell, and a receptor. And these parts work together in predictable steps: reception, transduction, and response, and then shut it all down with termination. Along the way, receptors and relay molecules change shape between active and inactive forms by changing conformation. Conformation and shape mean the same thing. Now, there are families of signaling pathways based on shared characteristics, and the one that we looked at today is G protein linked reception. And G protein signaling uses a G protein, GTP, and GDP that results from hydrolyzing that GTP, and that acts as a molecular switch to turn on the G protein, which then activates an enzyme and causes a response in the cell.